Welcome to this video where I look at nucleic acids including DNA and RNA. Let's first look at the structure of a nucleotide. This is the simplest repeating unit which makes up the DNA and RNA polymers. Here in this nucleotide we have three components. The first component here is the phosphate. The complete structure is shown. In this five-membered ring, this sugar, this pentose, is a deoxyribose if it's DNA, and it's a ribose if it's RNA. And a deoxyribose has a H attached as shown. If it's a ribose sugar, instead of a H, it has an OH. So deoxy means lacking oxygen. Now the way this pentose, this ribose or deoxyribose is numbered is as follows. There's a carbon which is hidden here, 5' carbon. The one here is 4'. The prime indicates we're labeling the carbons on this pentose. We have 3' carbon there, 2' and 1'. And it's a 2' carbon which is going to be lacking the oxygen in DNA. Now I'm going to come back to the 5' carbon and the 3' carbon. Notice the 3' carbon has a free OH group, which is very important when forming nucleotide bonds. The final part is the nucleotide base. This is attached to the 1' carbon. And this is, in this case, the base is adenine, but it could be another base, and I'll come to that later. So in summary, the phosphate is attached to the 5' carbon. The 3' carbon has a free OH group. The 1' carbon is attached to the base. The base, you will see, is rich in nitrogen. When making or synthesizing DNA and RNA, you need a source of nitrogen. For example, when plants photosynthesize, they make glucose. The glucose can combine with nitrates from the soil, which is a source of nitrogen, and this can make nucleotides. At A level, we draw nucleotides using a simpler notation as shown. Now here, the pentose, shown here, is very important to refer to it as a ribose or deoxyribose, depending on whether you're looking at RNA or DNA nucleotides. So how do nucleotide bonds form? Well, here on the left hand side I have two nucleotides. You can see that they are RNA nucleotides. They are ribonucleotides because they have a ribose sugar, because the 2' carbon has this OH, it's not H. Now I'm actually interested in the OH on this 3' carbon here on the left. That OH of this nucleotide will form a nucleotide bond with the phosphate of the next nucleotide. So in this way, the nucleotide can be bound to an adjacent nucleotide. Now you can see that the OH and on the 3' of this ribose and the OH in the phosphate. Okay, if we look at those, we need to lose water from them in order to make this bond. And indeed, we lose water in this condensation reaction. And we only retain one of those atoms, the oxygen. So it's important also to understand that if we wanted to add another nucleotide to this dinucleotide, we would have the phosphate, this phosphate, go and react with the OH of this carbon, and we will extend the chain this way. So here it's very important to understand that when DNA or RNA is synthesized, it has directionality. Now you can see here there's a 5' carbon. That is a carbon which is at the beginning. At the end of this chain, or this dinucleotide, we have a 3' carbon. And this is why we say the direction is 5' here to 3'. So if this was a long molecule with many nucleotides, then at this end it would be 5', at the bottom end it will be 3'. And so we're moving, as we're going down, we're moving from 5' to 3'. It's important to understand this directionality, as enzymes such as DNA polymerase can detect the 5' and 3' ends, and it moves in a certain direction. The nucleotide bond shown here is also called a phosphodiester bond, because this COP bond, going from this C to this P, this bond here is a single phosphoester bond. The bond underneath is also a single phosphoester bond. Because they are two, we say it is a phosphodiester bond. It's also called a nucleotide bond. Bases we can have in nucleotides in DNA are adenine, guanine, cytosine, and thymine. In RNA, instead of thymine, we have uracil. So it's uracil, not thymine, in RNA. But in DNA, we have one of these four in the nucleotides. Now, adenine and guanine, you can see, are made up of two rings. So in adenine, we have this ring here, and then another ring, and same for guanine. And this makes these nucleotides purines. Cytosine and thymine have a single ring each. These are pyrimidines. Uracil is also a pyrimidine. It's also important to understand that bases in nucleotides can complementary base pair to specific bases. So A in one nucleotide will be able to complementary base pair to T in another nucleotide. If we have the situation of RNA, then A in one nucleotide can complementary base pair to U in another nucleotide, because U takes the place of T in RNA. G can complementary base pair with C. And so we can see that on the left we have a purine, and on the right we have a pyrimidine. Purines complementary base pair to specific pyrimidines. Also, you will notice that the bonds between A and T, I've shown two dashed lines. These are hydrogen bonds. A and T complementary base pair by forming two hydrogen bonds, and it's the same for A and U. Whereas G and C complementary base pair by forming three hydrogen bonds. So it's a much stronger interaction here between G and C. On the left, you can see the well-known structure of DNA, the double helix. I've shown some dimensions. So the width of DNA about 2 nanometers. The length of DNA when it completes one turn of the double helix about 3.4 nanometers. And this is a specific type of DNA which we normally find in genetic material. So you can see the DNA is made of sugar phosphate backbones. There's two of them, and these are held together by complementary base pairs between bases on opposite strands. Now, the way you're going to see this drawn at A level is on the right hand side here. Here we have untwisted the DNA, and we've shown the components in a very simple format. You can see in green we have the phosphate. Because it's DNA, this orange is deoxyribose. This is one strand, and the bases are pointing inwards. And then we have the opposite strand. And the two strands are held together by these hydrogen bonds between the complementary base pairs. So here we have A, on the right hand side we'll have T. Here we have G, on the opposite side we'll have C, T, and then on this side we'll have A, and then C, the opposite side we'll have G. Another important thing to note is that if we have double-stranded DNA, we can infer a pattern when we count the total number of bases. So if we look at A, 2 of these bases are A. And because the total number of bases in this very small, short section of the molecule is 8, we can say that a quarter or 25% of the bases are A. It therefore follows, because A complementary base pairs with T, there'll be an equal number of bases which are T, which are 2 T bases. We have one here on the top and one on the third row down. So the number of the percentage of A, the percentage of T are equal. The same can be said for G and C. We have 2 G's and therefore we'll have 2 C's. The percentage of G equals the percentage of C. So in summary, in double-stranded DNA, the percentage of A, or the number of A bases that are A, will equal the percentage of T. And the percentage of G will equal to the percentage of C. If we have a single-stranded DNA molecule or single-stranded RNA molecule, we don't see any pattern. The percentage of A, T, C, and G will not equal each other. Finally, you can see, as I mentioned before, there's a directionality in DNA. Let's look at the strand on the left here. Now, the carbon at the top here is 5'. The carbon at the bottom, if you ignore this, let's say that this is the last nucleotide in this sequence, carbon at the bottom will be 3'. So moving from there, from the top to the bottom, is 5' to 3'. And you can easily understand this. An easy way of understanding this is if this ribose is pointing upwards, then the 5' end of the DNA is in that direction, right at the end. So it's up here. Now you can see on the left-hand side, the backbone is pointing downwards. So the ribose is pointing downwards. Okay, and this tells us that the 5' end is below here. And this means the 3' end is going to be above there. The 5' to 3' is going in this direction. So you can see the direction of the DNA strand on the left is in the opposite orientation to the direction of the DNA strand on the right. So on the one on the left, it's going from 5' to 3' downwards. On the right, 5' to 3' upwards. This is called an antiparallel arrangement. DNA is synthesized in the direction 5' to 3'. So this strand in red is 5' on this side, and it's being synthesized that way. The strand at the bottom here is 5' on this end, and it's being synthesized this way because the DNA polymerase is moving that way. It therefore follows that the template strand in blue at the top here is 3' on this end because of the antiparallel arrangement. And then this bottom strand here in blue, this template strand, is going to be 5' at the end here because it's an antiparallel arrangement. Okay, so let's have a look at what happens. Number 1, DNA helicase binds DNA. Number 2, DNA helicase unwinds DNA by breaking hydrogen bonds between complementary base pairs. Number 3, free nucleotides base pair to complementary bases on the single-stranded DNA, and you can use this abbreviation ssDNA if you wish. Template. So here what's happening is the helicases is around the DNA template. We have single-stranded DNA. You can see 3 nucleotides here. What they do is they complementary base pair, or the hydrogen bond to complementary bases on the template DNA. And it's going to be the same for the template DNA at the top here and the template DNA at the bottom here with the 3 nucleotides on the complementary base pair in this direction. Number 4, DNA polymerase. Forms nucleotide bonds between the 3 nucleotides that are bound to the template. So once this DNA here, let's have a look at this nucleotide. Once this nucleotide, complementary base pairs here, for example, then the DNA polymerase will form a nucleotide bond between this nucleotide and the growing DNA strand here. And so here, all of these nucleotides in red here, they were free nucleotides. The complementary base pair to the template, and DNA polymerase moved across and it just joined them together through nucleotide bonds. And now you see can form a new strand, and that new strand just grows as you move along here. So this results in a new strand of DNA. So it's quite straightforward what's happening, and it happens for both strands. Now, DNA's replication is semi-conservative in nature. So what does this mean? Well, semi-conservative means that, firstly, each DNA strand in the original DNA acts as a template to synthesize a new strand. And secondly, each resulting DNA after replication contains one initial strand and one new strand. So let me show you with the aid of this picture. So this parental DNA contains 2 original strands. Original is in purple here. Now, when the 2 strands in this double helix, when they unwind to give single-stranded DNA, 2 single purple strands, the single purple strand here on the left is used as a template to make a new strand, which is shown in blue. And so this DNA molecule here, which is now a new DNA molecule, it has an original purple strand and a newly synthesized blue strand. And it's the same for the case for this DNA molecule here. And if you repeat the cycle of replication, so we're going from there now to here, you can see that this strand in blue has been used to synthesize a new blue strand. And you can see both strands in this molecule. This pink strand has been used to synthesize a new strand. And you can see both strands in this DNA molecule here. And you can see the same is the case on the right hand side there. It's also important to understand that each time you replicate, you double the number of DNA molecules. Okay, so how has this been proven? What experimental evidence did scientists use to show that semi-conservative replication occurs in organisms? Well, the first point is that If we have DNA in bacteria, we can label the DNA or incorporate two different types of nitrogen in the DNA. One is nitrogen 15 and another is nitrogen 14. So if we grow bacteria on medium and put in nutrients that contain nitrogen 15 in the DNA, then the bacteria will take up the nitrogen 15, and the DNA it will make and the bases that it will make will contain only nitrogen 15. If we give growth supplements and nutrients to the bacteria which contain nitrogen 14, the only nitrogen it will incorporate into its DNA is nitrogen 14. Nitrogen 15 is heavy. It has a higher relative atomic mass than nitrogen 14. So a DNA that contains nitrogen 15 is going to be heavier, more denser than nitrogen 14 DNA. So here in purple, let's imagine the bacteria have been grown for a long time over multiple generations on nitrogen 15. It will have a heavy DNA. And then, so if we have a centrifuge tube like this, and in the centrifuge tube we have a cesium chloride CaCl gradient. All this is, cesium chloride is a salt, and we can make this tube so that we have a solution of cesium chloride, and the concentration of cesium chloride is higher at the bottom and lower at the top. This makes the solution more dense at the bottom and less dense at the top. If we now add the DNA onto the top of this tube, we centrifuge it in a very fast centrifuge called an ultracentrifuge. The DNA will move down and it will stop when it encounters a solution of equal density. So this DNA, which is N15, when you centrifuge it, it'll move down right to the bottom because it's heavy. Now, what we do is when we go from this parental DNA to the next stage, we grow the bacteria on not N15 but N14 nutrients, and so we allow the bacteria to grow for one cell division. We only allow one cell division, which means there's only one round of replication of the DNA. And what happens there is that the original N15 DNA is used as a template to make new DNA. That new DNA in blue is going to be N14. So the N15 DNA is in purple, the blue is N14. And you can see, so we get a hybrid DNA. This DNA molecule has one N15 strand, one N14 strand. It's not going to be as heavy or as dense as the DNA, which is the parent. It's going to be slightly lighter because this N14 DNA, which is complementary base pair to the N15, makes it slightly lighter. So it's going to have intermediate density. And so I'm going to put it up here like this. That's where the band will be. I've also increased the thickness of this band because we have double the amount of DNA. We've gone from one molecule to two molecules. Sometimes they don't worry about the thickness at A level, but it's worth knowing. Now think about what's going to happen here in this experiment. So we have two double helices which only have N14. We have this one here and that one there. The two molecules which have only blue strands. Now, because both strands are light, that's going to be a light DNA molecule. It's going to be right at the top. Now, we also have one which is similar to the one we had before, intermediate density. We have two of those molecules which have a purple and a blue strand, which is a light and heavy strand. So intermediate density. There we go. And my drawing isn't going to be very accurate here, but in an exam they'll give an opportunity to make the drawing accurate, so it's best to use a ruler here. Also note that here after the first replication I have two molecules of DNA. At the bottom I have four. They are split. Two of them are intermediate, two of them are light. So the thickness of this band and this band are equal to each other, and the thickness of each is equal to the thickness of this band here in the second picture there. Okay, so this explains semiconservative DNA replication. So let's now look at the structure of ribonucleic acid, RNA, and there are three types of RNA molecules that we generally find in cells, which we want to discuss. Firstly is messenger RNA or mRNA, and this is a short sequence of RNA, and it contains codons. It contains the same information as the DNA sequence in a gene, and this is involved in translation. In other words, to make new polypeptide chains, new proteins. tRNA is transfer RNA. This tRNA brings amino acids to the ribosome for translation. The ribosome is a structure where translation occurs, where the sequence in messenger RNA is read, and this is used to make a polypeptide chain with a specific amino acid sequence. And we'll learn about this more in later videos. There are many different tRNA molecules. Each one brings a different amino acid. Then we have ribosomal RNA or rRNA, and this rRNA is associated with ribosomal proteins. So it is part of the ribosome, and it's a very important feature, and it plays a very important role in the ribosome. Looking at the structures of these molecules, mRNA has a linear structure. tRNA has what we call a clover leaf structure. So a clover leaf has generally 3 leaves, and this clover leaf structure in tRNA somewhat resembles the clover plant. You can see the structure here, and up here is where the amino acid attaches. Now this it's an interesting structure. We have regions such as here where we have no base pairing. So this region is single-stranded RNA or ssRNA. Then we have these regions here which are double-stranded regions of the RNA, and this is a particular feature of the tRNA molecule. We also have what we call an anticodon loop. And here we have the anticodon, which is a sequence of 3 RNA nucleotides next to each other, and they will complementary base pair to a specific codon in mRNA. And this will be dealt more in the video covering translation, which I will do later. Ribosomal RNA has a complex structure, and we don't need to discuss it because it's so complex. Now this leads to the interesting point about comparing DNA and RNA. I'm going to do this here. So let's compare DNA and RNA. And here we have to be very careful. We can compare DNA to specific RNA molecules, but I'm going to compare it generally to all RNA molecules. So DNA has 2 strands because it's a double helix. RNA has only 1 strand. The strand can be folded, it can have a complex shape, but it's only 1 strand. In ribosomal RNA, we can have different RNAs, but they are separate RNA molecules, and they each consist of one strand only. Also, in DNA, we have thymine, or T. In RNA, we have uracil, or U, instead of thymine. And I'm being very specific here, and I'm spelling out the point that uracil replaces thymine. DNA is a longer molecule, and the reason is because it contains many genes, for example, chromosomal DNA. RNA is generally a shorter molecule, and the reason is because RNA is encoded by a single gene. Finally, DNA has a deoxyribose sugar, and RNA has a ribose sugar. Now, I haven't said that RNA is single-stranded. In particular, tRNA and ribosomal RNA have regions which are double-stranded. But if I was comparing DNA with messenger RNA, I would still not refer to the mRNA being single-stranded. The reason is because mRNA, although it's a straight molecule, generally, it can have something called hairpin structures, like this. And you can see that this stem here is a region which is double-stranded RNA, and this loop here is single-stranded. So mRNA can form regions which are double-stranded. Now let's go beyond the spec. Okay, there are 2 points I want to look at. The first one is looking at DNA replication in more detail. In fact, DNA replication is a complicated process, and it has been simplified at A level. The first point to understand is that there is a large protein complex containing many different proteins which are involved in replication. The first protein that binds to the DNA, the double-stranded DNA, is called the DNA initiator protein. It binds to a region of DNA called the origin of replication. That is a site on the DNA from which replication starts. In bacteria, the DNA initiator protein, an example is DNA-A. After this protein binds, it can help other proteins to bind. The next protein to bind is a large complex called the pre-initiation complex. It contains many different polypeptides. After this, DNA helicase binds, and this unwinds the DNA. Because the DNA is being unwound, the region of DNA which is now single-stranded, it could actually form double-stranded DNA again. There's nothing stopping it from complementary base pairing to each other, and this is an issue. To avoid this problem, single-stranded DNA binding protein will bind to DNA templates which are single-stranded. Any region which is single-stranded, because these proteins are bound, they will not be able to form complementary base pair with the opposite DNA strand. It will keep them single-stranded until the DNA replication has had a chance to copy the DNA. Another important point to consider is the DNA clamp. The enzyme that replicates DNA, as we mentioned, DNA polymerase, it moves along large stretches of single-stranded DNA, and this can often mean that it has an opportunity to fall off. It can fall off. To prevent this, a DNA clamp protein holds the DNA tightly, and it prevents DNA polymerase from falling off. Topoisomerase. So here on the right hand side you will see a double-stranded DNA. This is circular. This is like a bacterial chromosome. Now normally DNA, because we want it to occupy a small space inside the cell, we can supercoil it. And this is an example of supercoiling. Basically, the DNA is twisted, and this twist is a permanent twist. It cannot be unraveled by itself. It needs enzymes to unravel it. An enzyme that does this is called topoisomerase. It reduces the supercoiling. So, for example, instead of having 4 twists, I might just have 2. That means there's less supercoiling. And because the DNA is twisted, this makes it more difficult to replicate the DNA. For example, it makes it more difficult for the helicase to unwind all the regions of the DNA. And if we relax the DNA, then it can get less supercoiled, then it means that it's easier for replication to occur. Another important enzyme is DNA gyrase. Now let's imagine we have double-stranded DNA here. I have 2 single-stranded regions on the left-hand side. And DNA helicase here is moving down this double-stranded DNA and it's unwinding it. So here we have unwound regions. As the helicase moves down, it can cause strain on the region of DNA which is not yet unwound. To reduce the chances of this, DNA gyrase will relax the DNA which is not yet unwound, and this will allow helicase to proceed at the same rate along this DNA. DNA ligase is an enzyme I'm going to come to on the next slide. So DNA replication is a complex process. Here we see a site here on this double-stranded DNA called the origin of replication. And that's where replication has started. Now, when you replicate DNA, it doesn't just go in one direction. It goes in both directions, bidirectional. And so, starting from this origin of replication, we can either replicate the DNA moving right or moving left, and both of these happen at the same time. Now let's look at one half. Let's look at the right side of the origin of replication to make it easier. Now the template DNA is shown in purple, the one at the bottom. Let's have a look at this one first. Now you can see that owing to the antiparallel nature of double-stranded DNA, the newly synthesized DNA, which is shown here in yellow, is being synthesized 5' to 3' in the direction of the arrow, like this. In order to do this, we need to first tell DNA polymerase where to start replication. And so an enzyme called primase will synthesize this primer. The enzyme that makes it is called primase. This primer is a short sequence of single-stranded DNA or RNA and is complementary base pair to the template at a particular site. Now at this end, at the 3' end of this primer, we have a free 3' OH group. And so when free nucleotides, such as this, when they complementary base pair to the template, the DNA polymerase will form a nucleotide bond between the 3' OH of the primer and this free nucleotide, as it has already done so in this picture. Now, the 3' OH group, the free 3' OH group will then be at the end of this nucleotide, because at that point that will be the end of this molecule. And now we just add nucleotides on, and we just move on. We move from 5' to 3'. And this free 3' OH on this primer tells DNA polymerase where to start replicating. Now, this strand here is called the leading strand because we're replicating from 5' to 3'. At the top here is a different story. The replication fork and DNA helicase are moving this way. We saw the leading strand, the DNA is being synthesized in the same direction as a replication fork, and so it has no issues. At the top here, this purple strand, this template strand, when it's copied, the newly synthesized strand is moving in the opposite direction to the replication fork or the DNA helicase, it's moving in the opposite direction to the direction that the DNA is being unwound. And so this is an issue because we're moving in the opposite direction. And I want to show this to you in a bit more detail. So let's imagine that we have double-stranded DNA here. Okay. Now let's imagine this region of the DNA has not been unwound, and the replication fork, or this structure where we have single-stranded DNA and then next to it double-stranded DNA, which is called replication fork, this is moving that way. So DNA is being unwound in this direction. Now let's say we're replicating the DNA in this direction. So let's say this region has been replicated. This is my new strand. Let's now unwind this region here. So we've just unwound a bit more of the DNA. Here is my newly synthesized DNA, but now you can see this region of the DNA has now been unwound, and now we need to replicate it. So what I need to do is I then need to go back to here and synthesize a strand. I can't synthesize a continuous strand because I need to synthesize a strand from this end, because this end is going to be 5' on the newly synthesized DNA. But that region of the DNA has not been exposed, it's not single-stranded. So I have to start here. So I have to synthesize these short strands of DNA, and these are called Okazaki fragments. And so this is called the lagging strand, and the DNA synthesis on this strand is much slower because we're synthesizing DNA in the opposite direction to the fork movement, and every time more single-stranded DNA template is exposed, we synthesize another short sequence. To do this, we need multiple primers. We need to tell the DNA polymerase to start here, and when more of this double-stranded DNA is unwound, we're going to start later there. In order to then join the gaps between these DNA strands, what happens is something called DNA ligase forms nucleotide bonds between these short sequences, these Okazaki fragments, and this then leads to a continuous long strand of DNA. I hope you enjoyed watching this video. Please also check my other videos on the website.